All right, so we are on chapter 10, section 4. We're doing what happens to proteins when things go wrong, and that's denaturation of proteins, right? So we know that we have attractive forces that stabilize our protein because of secondary, tertiary, and quaternary interactions, right? And so all of those attractive forces stabilize this one protein and hold it all together. So when you denature a protein, sometimes it's heat, right? Like when you cook an egg, what happens is that protein that normally has alpha helices and beta sheets and all this kind of stuff just kind of ends up as a spaghetti noodle. Right. And when it turns into that spaghetti noodle, instead of looking like the telephone cord and the fan and this and that and folded this way and that way, it turns into a spaghetti. It doesn't work anymore. And so it loses its biological activity. So it could be heat, it could be changing the pH, so adding an acid or a base. You can add heavy metal salts. You could just agitation, just putting it in the blender, right, can interfere with those interactions and end up denaturing the protein. And so if you don't have those stabilizing interactions, you don't maintain the three-dimensional shape, and you got this string or this spaghetti strand of amino acids, and that's it. So here are the examples of the different denaturing, excuse me, denaturing agents that can interrupt three-dimensional shapes of proteins. So if you have something that is above 50 degrees Celsius, well, then that's going to break hydrogen bonds. That's going to disrupt hydrophobic interactions. Well, that's what we do when we cook every day. You can add acids and bases. If you add acids and bases, you change the pH. If you change the pH, now things that were positive and things that were negative are no longer positive and negative, right? So this happens when we make yogurt and we make cheese. We start with milk, and when we incubate it with bacteria, the bacteria produce lactic acid. The lactic acid then affects the proteins in milk and turns them into yogurt and cheese, and they're delicious, right? All right, reducing agents, right? We talked about that, things that will remove those hydrogens, right? So disulfide bonds, they'll disrupt disulfide bonds, and we talked about that with our thiols and hair straightening and waving. So the same thing, I use the example in the lecture about making curls permanence, but you can also do the same thing with somebody who has curly hair who wants straight hair, right? You can chemically treat it, and then you straighten it, and then you reduce it back to the original form. So then they're locked in that straight orientation. Detergents, so if you want to break up things like cell membranes, right, you can do the same thing to proteins. If you add a detergent, you're going to disrupt the hydrophobic interactions. And so that will really mess up membrane proteins. Ions will mess up disulfide bonds and salt bridges. And so that's what happens if you have mercury or lead poisoning. It's very, very dangerous. And then mechanical agitation. So if you are going to make a meringue, right, from your egg whites, you're going to put it in the mixer and beat them until they get stiff peaks, right? So what are you disrupting? You're disrupting hydrogen bonds and London forces. So hair relaxing, this is the same sort of thing I was talking about last time, right? And in terms of perms, it's the same sort of deal, but in reverse. So if you start out with curly hair, you have proteins in your hair, the keratin, that have those disulfide bonds. And so they're locked in this orientation where they're very, very nice and curly. And so if you add a reducing agent, what happens is that those disulfide bonds break. And so now you end up with thiols, right? So we have a bunch of thiols. And then you rearrange the hair. So you make it very, very straight. And then while it's still straight, you reoxidize those disulfide bonds inside of the keratin, and now you have straight hair. And you could go in reverse, right? You could start with straight hair. You could reduce it, then put it in a curler, then oxidize it, and it'll stay in that new shape, right? So denaturing proteins is really, really important in terms of treating different things, right? So if we know that someone has had lead or mercury poisoning, right, we can give them egg whites to drink, and the egg whites are denatured by the lead or mercury and form a precipitate. And then you would pass that precipitate in order to get rid of it. And the quickest way to do that so that you get it out of your body is to induce vomiting. And so you give the person egg whites, it makes that precipitate, and then you make them vomit, and then you get rid of it. So it's a really quick way to get rid of somebody who has ingested lead or mercury. All right, so now we're going to switch to a couple of quick questions. All right, so I put in your little table so that it would be really quick and easy to find, where we have examples of our protein denaturation. So list the type of attractive force that is disrupted and the level of the protein structure changed by the following denaturing treatments. Adding salt to soy milk to make tofu. So remember, if you're adding salt, salt is an ionic compound. So this is what we're doing. So what are you going to disrupt? You're going to disrupt salt bridges, and you're going to disrupt hydrogen bonds. So now the question is, what secondary structures are you going to interfere with? If you're interfering with hydrogen bonding, you're interfering with hydrogen bonding between side chains and hydrogen bonding in the backbone. So that tells me that I have both secondary and I have tertiary interactions. And we know that salt bridges are really important for tertiary structures. And so if you lose secondary and tertiary structures, guess what else you lose? Quaternary structures. Whipping eggs into stiff peaks to fold into an angel food cake. Sounds great, right? Mechanical agitation, that is what you are doing to it. So what are you disrupting? Hydrogen bonds, and we're disrupting London dispersion forces. Well, hydrogen bonds, that's telling me secondary, quaternary, secondary and tertiary. And if you're disrupting secondary and tertiary, you're disrupting quaternary, right? And the London dispersion forces are in tertiary and quaternary, right? Okay, identify each of the statements as characteristics of protein denaturation or protein hydrolysis. So remember, denaturation is just turning it into a spaghetti. It's all about food, this one. And protein hydrolysis. Hydrolysis is when we are actually cutting up our amino acids. So we're going from one long chain and we're turning it into, right, some little bitty ones. So you're actually chopping it up. You're breaking covalent bonds. And those covalent bonds are the peptide bonds. Okay, let's see. Milk curdles when lemon juice is added to it. Lemon juice, you got to remember, is very, very acidic. So if you are adding, see, acidic. So if you are adding an acid, you are denaturing. You're not actually cutting it up, but you're just turning it into spaghetti. Protein breaks up into amino acid fragments. Oh, hydrolysis, right? Because you are breaking it up into amino acid fragments. And I think, yeah, that's it. What a nice short one. Have a great rest of your day.